Hello viewers, welcome back to my channel Knowledge Group. And first of all, let me tell you one thing, that if you need these in soft form, you can contact or mail me. I'll paste my mail account in the description box, and you can also comment me and press the bell icon and subscribe to my channel. So let's start. The structure of Eubacteria. Basically, it's a type of bacteria, and you may know that because you are all very intelligent. So, the structure of bacteria contains cell wall, cytoplasm, a nuclear material, and flagella, and the last one is the mesosomes. So, the next, cell wall. It is a cell wall of the bacteria, but not the bacteria. It's a type of bacteria that is Eubacteria, means it has true structures. It has true structures. That's why it's known as Eubacteria, means they're recent or advanced bacteria, not the Archaeobacteria, means they are ancient type of bacteria. So it contains strong and rigid cell wall which contain the muramic acids, and microscopy reveals that it's composed of granular units that is 50 to 140 nanometer in diameter, and the shape is regular, hexagonal or rectangular pattern. So basically their slimy capsule is composed of polysaccharides, and that slide these to the cell wall and serve as an additional protective layers. In the next, you can see the gram-positive and the gram-negative cell wall, and the gram-positive and gram-negative are discovered by the gram staining. Gram, it's a name of a scientist who discovered the gram staining technique. And the next is that here is the explanation about these gram-positive and gram-negative. Means that the gram-positive contain the one lipid plasma membrane, and that is thick peptidoglycan layer, while the gram-negative as a inner and outer membrane, and is a thin layer of peptidoglycan in the periplasmic space. So there is a layer between the lipoprotein lining and outer membrane of gram-negative bacteria, not in the gram-positive. The next is the glycocalyx and capsule or slime. So what is the glycocalyx? Basically, it's a sticky substance that surrounds outside the bacterial cell. It's made of polysaccharides and peptides, or both, like maybe some polysaccharides or maybe polypeptides. So what is capsule? Capsules mean organized and firmly attached to cell wall, but slime means unorganized or loosely attached to the cell wall. And it allows the pathogens to survive phagocytosis. Phagocytosis means eating of food. So as you can see in the Streptococcus pneumonia, and you can see in this diagram are Calbrecia pneumonia and Bacillus anthracis. In the next, you can see the cytoplasm. Cytoplasm within the bacterial cell is a dense and contain granules and of the glycogen, protein, fat, and lack of mitochondria and endoplasmic reticulum. And it contains the RNA also. And ribosomes are free in cytoplasm and are not bound to the endoplasmic reticulum. And the cocci have one nuclear region but rod like bacilli, and usually have two or more nuclear regions per cell. In the next, you can see the nuclear material. Nuclear material means fifth portion out of one is the interior of bacteria cell occupied by the nuclear material, and it lacks the nuclear membrane and nucleolus, and is a prokaryotic. Prokaryotic means do not organize nucleus, and they exist a low electron density which is occupied by DNA, the genetic material, and is a region known as nuclear body or nucleoid. And the nucleoid is associated with the mesosomes of gram-positive, and the DNA strands is equivalent to chromosomes. In example is Escherichia coli. And the chromosomes have identical genetic composition because of the product of same parent chromosomes. In the next, we will discuss mesosomes. In some bacteria, Bacillus subtilis, small circular, semicircular bodies occur attached to plasmamilla. And this body called mesosomes. Mesosome consists of coiled membranes, and they induce divisions of nuclear body for the formation of septa. As you can see in this diagram, there you can see the cell wall, the central mesosome, and the peripheral mesosome. So in the next, with the flagella. So wave-like flagella outgrowth called flagella have wave-like outgrowth and can swim about. And they consist of single fibrils where flagella of higher organisms composed of 11 fibrils and bundles and 9 in peripheral. You can see 9 and 11. 9 and 2 in the center. So rod shaped and spiral shaped bacteria and have the flagella, whereas spherical ones do not. And the flagella and not flagellates form called arthropods. So you can see the structure of bacteria, you can see the pili, the capsule, the outer membrane, the cell wall, the glycon layer, plasmid, and DNA that's present in it. And one important is the flagella. Bacteria has a flagella. So you can see the pili. Pili is that here is the example of circula. You can take the example. It's a very short and narrow and flagella-like numerous protoplasmic outgrowths of all around the body. And the outgrowths called pili. Okay, and the pili are the protein structures and they are existed about 2 micrometer, and the circuli have 300 of these organelles, and they constricted and structure of protein and pylences. You can see it's a definition of pylens. And straight slender and fibrils are similar to flagellum, like they are structurally similar, and they have the attachment of the water surface and the formation of irrigation soil product and during their germination. So in the next you can see the forms of the flagella. The first one is the atrichos, and the next is the monotrichos and amphitrichos. Means that not flagella. Mono means only one flagella and amphi means one flagella at each end. But in the other, here is the cephalotrichos, lophotrichos, and peritrichos. Means that group of flagella present, tuft of flagella at one end. Here you can see one end. And peritrichos mean all over the body. That you can see in this diagram: atrichos, monotrichos, amphitrichos, peritrichos, lophotrichos, and cephalotrichos. So movement of flagella. Now we'll discuss. The movement is very unique. It's not the vibe-like movement, okay, and it's a root like just like wheel or perform the corkscrew movements, and their number is different from species to species, and they lack the flagella, and movement is a snake-like movement, and some is to travel to 2,000 times, and their own length 1 hour with the help of flagella. And in this picture, types of rotation by flagella and you can see there is a reverse flagellation, there is a unidirectional flagellation, and the polar flagellation. And just CW, you can see in this diagram very clearly. You can see that. And thanks for watching. If you like my video, then comment me or press the bell icon for more videos, for more subscription. And if you need in the soft form, you can ask me. I'll paste my mail account in this description box. Thank you very much.